Okay, so let's dive right in. We all know how incredibly complex the brain is, right? But what actually happens inside, at a microscopic level, when something goes wrong? Today, we're going to pull back the curtain and look at the intense cellular drama that unfolds the moment the brain gets hurt.
I mean, it's a huge question, isn't it? This 3-pound universe in our heads that holds all our thoughts, our memories. What happens when it takes a hit? You might just imagine total chaos, a system in complete meltdown, but that's not really the full picture.
In reality, the brain's response is this highly organized, almost military-like operation. It's a rapid deployment of a specialized team of cells where every single one has a very specific job to do. So, to really get what's happening, first, we've got to meet the team.
And the most important player on that team, the one the entire system is built to protect, is the neuron. You can think of it as the brain's lead actor, its very fragile superstar.
Every single thing you think, feel, or do is because of these cells. They're the wiring, they're the processors, the memory banks, all of it rolled into one. But for as important as they are, they have one massive critical flaw.
And this right here is the whole ballgame. Unlike your skin cells, which can heal, or your liver, which can regenerate, once a mature neuron dies, the brain can't just make a new one to replace it. It's gone. This is exactly why brain injuries can have such permanent, devastating consequences. The stakes are just incredibly high for every last one of these cells.
So since they're so precious, it's really important to understand how they get damaged. And generally, you can break down neuronal injury into two main types, and it all depends on how fast the damage happens.
On one side, you've got acute injury. The classic example is a stroke, cutting off blood flow. Within just 12 to 24 hours, the neuron shrinks up, its nucleus gets all condensed, and it stains this intense red color under a microscope, which is where it gets the nickname red neuron. This is irreversible. It's done. Then on the other side, you have the slow burn of chronic injury, like what you see in diseases like ALS. Here, neurons die off slowly, gradually, over a long period of time. It's a much more insidious process.
Now this is where it gets really interesting. A lot of these injury processes, they leave behind clues, little bits of abnormal stuff inside the neuron called inclusions. And they're basically like pathological fingerprints. Alzheimer's, it leaves behind neurofibrillary tangles. Parkinson's has its famous Lewy bodies. Even viruses like rabies leave their own distinct calling card inside the cell.
So, a neuron gets hurt. Who's the first on the scene? Meet the astrocyte. If the neuron is the superstar, the astrocyte is the tireless first responder and the site manager. And its reaction is the single most important sign that something has gone very wrong. And this quote right here is key. What it's saying is that it doesn't matter what caused the injury. It could be a stroke, physical trauma, a disease. The very first and most reliable sign that pathologists see under a microscope is something called gliosis. This is the astrocyte's signature move.
So what is gliosis? Well, in the simplest terms, it's the brain's way of forming a scar. The astrocytes near the injury, they get bigger, that's hypertrophy, and they start to multiply, that's hyperplasia. They swell up, their insides turn pink, and their main job is to wall off the damaged area, to create a protective barrier.
But just walling off the damage isn't enough. You also have to clean up the mess, and that's where our next player comes in. Enter the microglia, the brain's dedicated cleanup crew and its personal security force. Yeah, so unlike most other brain cells, microglia are actually part of the immune system. They're the brain's own resident macrophages, which is a fancy way of saying their job is to find threats, clear out all the debris, and, yes, literally eat dead or dying cells. And they have a very specific playbook they follow. First, they multiply. Then, they change their shape. Their nuclei get all long and skinny. They start clustering together in what are called nodules around dead tissue. And finally, they do their most critical job, neurophagia. It sounds dramatic because it is. It's the process where microglia swarm a dying neuron and just devour it, removing it before it can cause even more damage to its neighbors. Now, of course, there are other supporting cells. You've got oligodendrocytes, which make the myelin insulation for neurons, but in the immediate active response to an injury, it's really the astrocytes and microglia who are the main characters of the story.
Okay, so we've met the team, the fragile neuron, the first responder astrocyte, and the cleanup crew microglia. So let's put it all together and see how they actually work during a real-world crisis like a stroke.
The sequence of events is amazingly predictable. In the first day, boom, the red neurons show up.
Within about 1 to 3 days, immune cells from the blood called neutrophils rush in. But by about day 10, the microglia are totally in charge, busy cleaning up all the debris while the astrocytes are hard at work building that glial scar. Eventually, all that dead tissue gets cleared out, leaving behind a little fluid-filled cavity that's walled off by that dense scar.
So that's the story for a big sudden injury, like a stroke. But what about those chronic diseases we talked about, like Alzheimer's and Parkinson's? Well, they're driven by a different, much more subtle kind of problem.
These diseases are often called proteinopathies. It sounds like a complicated word, but the concept is pretty straightforward. It's a disease caused by proteins that are just behaving badly. See, proteins have to fold into these super specific 3D shapes to do their jobs. When they misfold, they get sticky, they start clumping together, and those clumps become toxic to the neurons around them. And this right here brings us to one of the most critical insights in all of modern neuroscience. For decades, scientists thought the big, visible clumps, like the amyloid plaques you hear about in Alzheimer's, were the main problem. But it turns out we were looking at the wrong culprit. The real damage is caused by tiny, invisible, soluble clumps called oligomers. These are the real killers that mess up how neurons work long before those big plaques ever even form. So this leaves us with the crucial question, right? If these tiny, invisible oligomers are the real villains of the story, can we find a way to stop them? Can we clear them out before they kick off that devastating cascade of damage? Answering that question is really the great challenge, and honestly, the great hope for the future of treating these devastating brain diseases.